Welcome students to my channel Learning Made Easy. I am Roxana Marks, PGT Biology and an ex-principal. Today we will be talking about the next thing that I left in the first video, that was the structure of pentoses and hexoses. As we discussed there, these are the two main important sugars which are used in our biomolecules. That means molecules present in life. Now the first one is pentoses. When we are talking about pentoses, in the first video I did tell you that pentoses are of two types. One is ribose and the other is deoxyribose, a special structure. Now it has got two structures, as I did talk about in the first video, that they are in both forms, open chain form and a ring structure. Here we are first dealing with the pentoses. Pentoses, that means having 5 carbon. Now here is the chain structure of pentose. I have numbered the carbon. This is to understand the position of OH and H easily. You can omit them while you are doing your examination papers, but this is just to understand that the numbers given to the carbon, where they are located and which of the groups are associated with them. So here in pentoses we find that the first carbon is joined with a double bound O, oxygen, and a hydrogen on the other end, while the second and third carbon have an OH on the right hand side and H on the left hand side. The fourth also is similar. The fifth has got 2 hydrogen and an OH group. While dealing with the ring structure, this is a pentagonal ring, as we already dealt with them. Now this is a furanose ring having an oxygen on the upper end, then 4 carbons in the ring. These 4 carbons, the first carbon is attached to OH and H, the second and the third carbon, H on the inner end, OH on the outer end. Fourth carbon is associated with the fifth carbon, which is associated outside the ring, and the fourth carbon has an H on one end. This is ribose sugar. Now ribose sugar is the main backbone of RNA molecule, which is present as one of the nucleic acids. That is, prokaryotic cells have ribose in their DNA structures, in their genetic structure rather. There's a correction, in the genetic structure. While deoxyribose sugar is a part of DNA. Now this DNA is a special structure. In the first video I did say that this is a special structure. Now why is deoxyribose a special structure? Special structure is, it is placed under the pentose sugar but having 1 oxygen less as compared to ribose sugar. This sugar is making the backbone of DNA molecule along with phosphate molecule and has C5H10O4. That is, 1 oxygen is lacking out here, and that is why we call it as deoxy. And this sugar molecule is present in DNA. That is, the eukaryotic cells, they all have DNA structures as their genetic material. Ribose in the prokaryotic cell, while deoxyribose in the eukaryotic cells. Now the hexoses also form a major part of the living bodies. Now we have 3 forms of hexoses. All are isomers having C6H12O6. Now there is no special formula out here, only the ring structure is different, that will be taking up. Now the first one is glucose having 6 carbons. I will be numbering them. You need not number them. If you understand what I am telling you, you need not number them in your examination. Now the first carbon is attached to double bound O and an H. The second with an OH and H, the third with an H and OH, the fourth, fifth and sixth having OH on the right hand side, H on the left side, and having an H at the end of the chain. This is an open chain structure of glucose. While that of fructose is the first carbon has 3 hydrogen associated with it. The second carbon is associated with double bond O. The third carbon having an H on the right side, OH on the left side. The fourth carbon, the fifth carbon and the sixth carbon having OH on the right hand side, H on the left hand side. Now then comes the third structure, that is the galactose. The galactose is more or less like the glucose with just one difference in it. And this difference is, as you see, the first carbon, the second carbon and the third carbon, they are exactly like glucose. Now the fourth carbon, the OH position has altered. Now in glucose, it is on the right hand side, while on fructose, it is on the left hand side. So this is the major difference between galactose and glucose. All the three forms are called as isomers and they are placed under hexoses. Very important sugar for the human body. Glucose, fructose and galactose, the chain sugars are here, and as we have discussed, these are very important sugars for human body. Their ring structures are given here. Now the first ring structure is glucose. In this glucose, what we find is that the carbons have a little different positions for H and OH. As already discussed, we find that this ring structure is a pyranose ring. That means a hexagonal ring having 5 carbons in the hexagonal structure. The 6th carbon is outside the ring. Now the first has OH, H, then it is H, OH, OH, H, and the 4th carbon H, OH, and then the 6th carbon CH2 having an OH group. While the second one is fructose. The ring structure of fructose is a furanose ring. That means a pentagonal ring having an oxygen and 1, 2, 3, 4 structures in the ring, while the first carbon is placed here and the 6th carbon is present here. Now these carbon atoms, again, these carbon atoms are attached with one another in the ring, and this furanose ring is a sugar which is normally present in fruit. So sometimes we call fructose as fruit sugar. This is the third chain structure, that is galactose in the ring structure. Again, this is a pyranose ring having 5 carbon in the ring and an oxygen. Now here what we find is that the 4th carbon had a different structure. This is what we find, the 4th carbon of the chain had a different structure. So here also the groups are placed opposite to the glucose. So the structures here are simple and can be remembered easily with a little change in them, and these structures we will again be dealing when we will be talking about their functions. And then thank you for watching. In the next we will be dealing about the functions of monosaccharides